Hi everyone, welcome to GemChem. Now today's topic of discussion is nucleic acids. Now before starting, if you are new to GemChem, do not forget to subscribe the channel and hit the bell icon for regular updates. So let us start. So in case of nucleic acids, generally we have two types of nucleic acids. One is deoxyribonucleic acid, that is DNA, and another is ribonucleic acid, that is RNA. And these respectively have the performance of preserving hereditary information and to transcribe and translate it in such a way so that synthesis of varied proteins of cell can be done. So, in our knowledge, we shall focus our attention into the different kinds of structure and properties of nucleic acid and their components. Generally, this DNA or RNA is made up of a monomeric unit which is known as nucleotides. Now, what is nucleotide? Nucleotide is the composition where there is a presence of heterocyclic base either in the form of purine or pyrimidine and a 5-carbon monosaccharide either in the form of D-ribose or 2-deoxy-D-ribose, and another is phosphate ion. So, these three together forms our nucleotide, and nucleoside is another component same as this one, but only the phosphate ion is absent in that case. So, we are going to see the nucleotide and nucleoside units for DNA and RNA. So, when we talk about nucleotides for RNA, this is our phosphate ion. Here we have the heterocyclic base, and this is our D-ribose which is present as furanoside. This is the sugar component, right? And we can name it as this one. From this carbon we can number as 1 dash, second carbon as 2 dash, third carbon as 3 dash, and fourth carbon as 4 dash. And this particular linkage between nitrogen and the carbon present, this is known as N-glycosidic linkage. Now, once we talk about nucleotides of RNA, we can easily construct the nucleotide for DNA just by replacing this OH present in the 2 dash carbon with a H, because it is 2-deoxy. So, oxygen is being lost, and we get our nucleotide for DNA as this one with hydrogen present in the carbon number 2 dash. Now, once we talk about nucleosides, we will see that this phosphate ion is being replaced by a hydrogen. So, the respective nucleosides for RNA and DNA are as follows. The structure remains the same, and here the part has become OH in nature. So, what are the nitrogenous bases basically present in RNA and DNA? 3 of the bases which are present in RNA and DNA are similar and same, whereas one of the base will be different for both cases. First, we are going to see adenine. So, you can see here we are getting a base with 2 rings. One is 6-membered ring, one is 5-membered ring, and here we have heterocyclic linkage. Another one is your guanine. So, guanine has the structure like this, where we have a double bond oxygen NH and NH2. These 2 bases are purine bases, and these same purine bases are present in case of DNA. No change occurs for these nitrogenous bases. So, these are the purine bases for DNA. Now, when we talk about another category, that is pyrimidine bases, in case of RNA we have cytosine and we have uracil. These 2 are our pyrimidine bases in case of RNA. But for DNA, uracil changes and there is a CH3 connected in this carbon, and this is our thymine. Cytosine remains as it is. So, the bases present in DNA is cytosine and thymine, and in thymine you can see that there is extra CH3. So, these 2 bases are different in case of RNA and DNA, and these are also pyrimidine bases. So, RNA nucleoside, uracil replaces thymine, and the heterocyclic bases obtained from nucleosides are capable of existing in more than one tautomeric form. The forms which we have seen here are predominant forms. That's why they are presented in this video. Now we are going to construct the nucleotides and nucleosides for RNA. So when we talk about adenine base with the carbon ring, that is for the sugar ring, along with OH, this is your adenosine. Once you add the phosphate group here, it becomes adenosine 5-phosphate, or known as adenylic acid, 5-adenylic acid, or adenosine monophosphate. Now next one, nucleotide and nucleoside for guanine is guanosine. Same structure, this part remains same, just the addition is the base, and the base is getting connected with this nitrogen, and this is your guanosine 5-phosphate. This is your cytidine, and this is cytidine 5-phosphate. Just the base is getting added here. If you consider the last one, uridine, and you have uridine 5-phosphate. Now when we talk about next case, that is DNA. The base is attached in same way, just the replacement is that the 2- carbon has hydrogen in it and the OH group has gone. Same case, this is for DNA. So here you will have in the same way, same structure, only while writing you have to be careful that H is being taking the places of OH in 2- position. So this is also for DNA. Next you have this one also. So in this case also, this is for 2-deoxycytidine, and here this is also one of the nucleotide and nucleosides for DNA. Here only the replacement is in this position. And the last one is thymidine. So thymidine 2-deoxythymidine or 2-deoxythymidine 5-phosphate. So in this case also it is for DNA. Right? Now what is the primary structure for RNA? That we should know. We will deal with the primary structure of RNA as well as primary structure of DNA. So in case of RNA, we can see that first we have a O connected, and this is our guanosine. Next we have cytosine. Next we have adenine, and last we have uracil. So this part is our 3- end. As you can see that here you have the 3- end, and we can write it or mark it as this is the 3- end, and this is your 5- end, and this is a hypothetical segment of RNA chain. Here you can see that this is sugar phosphate backbone, and there is a phosphate ester bond present. These bonds which are present here, as you can see here, these bonds are your phosphate ester bonds. Same way, if you try to construct the primary structure of DNA, only the difference will be instead of your uracil, you are going to get thymine, and the structure will have H in the deoxy position, that is 2- position, instead of OH group, and structure remains as it is. But DNA is a double helical structure, so when we are going to construct, there will be one chain running from 5- end to 3- end, and another will spiral up in the form of 3- end to 5- end. In this case also, this part is your 3- end and this part is your 5- end. So the base sequence helps us to encode the different genetic information, and techniques have been developed to get or determine the base sequences, and already those base sequences have been worked out for number of nucleic acids. Now our main important concern is what is the secondary structure of DNA. So classical model of DNA is being presented by Watson and Crick using X-ray analysis, and their secondary structure is especially important for different things, like it enables us to understand how genetic information is preserved, how those gets passed during cell division, and how it can be transcribed to provide a template for protein synthesis. So of prime importance to Watson's Crick's proposal was an earlier observation which was done by Erwin Chargaff, and according to his observations, the pointed out parts are, first is that the total mole percentage of purine is approximately equal to total mole percentage of your pyrimidines, and the mole percentage of adenine is equal to mole percentage of thymine, as well as the mole percentage of guanine is equal to the mole percentage of cytosine. And if you see this ratio varies from species to species. This is the main characteristics of a DNA of given species. It is same for DNA obtained from different tissues of same species, but once you change the different organisms, then this particular ratio changes. So what is the basic feature of Watson-Crick model? First is that DNA is generally constituting of two antiparallel strands. One is running from 5' to 3' direction and another is running in 3' to 5' direction, and they wind up in a right-handed manner across a common axis. Now the width of this particular chain sequence is 20 angstrom, and the bases are nearly perpendicular to the helix. It is at 90 degree to the helix, and this helical appearance generally repeats itself, complete helix, every 34 angstrom, and there are total of 10 bases in this particular 34 angstrom per turn of the helix. There is a rotation of 36 degree per bases, and each strand of the DNA has a deoxyribose phosphate backbone on the outside of the molecule, which we have seen, and these backbone forms major and minor grooves. So this is the minor groove, and here we can see there is a major groove, while the bases are present inside. This deoxyribose and phosphate units which we consider, which are present as a backbone, these are present on about the same plane, and the deoxyribose to purine and pyrimidine is approximately 90 degree. Thus, bases are projected at nearly 90 degree to the axis of the DNA. So the two polynucleotide chain are not actually identical, they are complementary to each other because the bases will attach in complementary pairs, and the two strands are held together by hydrogen bonds formed by these complementary base pairs. So how this is formed? There is a hydrogen bond formed between a purine and a pyrimidine only. So if we consider adenine and thymine, we are going to see that there is two hydrogen bonds taking place. Either adenine will bond with thymine or thymine will bond with adenine. In case of guanine and cytosine, we are going to see that either guanine will bond with cytosine or cytosine will bond with guanine. And here we have three hydrogen bonds present with each other. So the complementary base pairing in DNA helix proves the Chargaff's rule, that is, the ratio should remain constant for different species, and the mole percentage is also nearly equal to 1 for both cases. So the content of adenine equals to the thymine and guanine equals to the cytosine. So the genetic information is residing on one of the two strands, which is known as template strand and sense strand, and the opposite strand is known as anti-sense strand. And this is the main part which we have learned from this one. Nothing more is asked, but these structures are very important. So the secondary structure, when we try to go through the molecular model, it is just like this. There is a bonding taking place, and you can see here major and minor grooves are also present, and this is the molecular model of the DNA helix. So in this video we have dealt with the different parts of the DNA. We have dealt with the structures of different components present in the DNA, and here ends the topic of first part. Hope this was helpful. In the next video, we are going to deal with the different processes involved with the DNA, whether it is replication, transcription, etc. And in a short video, we are going to deal with how to synthesize these nucleotides and nucleosides in a laboratory. So hope this was helpful. Thank you for watching. Do not forget to like, share, subscribe and comment.